alaikum. Today we are going to discuss the histology of respiratory system, which will be divided into three parts. So today we are going to discuss part 1. The learning objectives would be that students should be able to understand the basic epithelium of respiratory system, understand the histological features of nose, nasal sinuses and nasopharynx, describe the histologic feature of larynx and epiglottis, and then you should be able to draw and label histological section of epiglottis. So the respiratory system consists of a conducting part and a respiratory part. The conducting part consists of nasal cavity, nasopharynx, larynx, trachea, bronchi, bronchioles, and up to the terminal bronchiole. It is the conducting part. The respiratory part starts from the respiratory bronchioles, then alveolar ducts and alveoli. So when the air comes in, it goes through all of these, which is the conducting part, and the respiratory part is the place where the gaseous exchange takes place. The functions of the conducting part is conduction of the air conditioning, temperature, moisture adjustments and cleaning. The nasal hairs which are present, also called vibrissae, they are used to clean and trap large particles. There are lots of capillaries to adjust temperature, and then we have the respiratory mucosa to adjust moisture. So it is basically the conduction of air and air conditioning through nasal hairs, capillaries and respiratory mucosa. The nasal cavity this hole is the nasal cavity. This is the opening of the nostrils, the nasopharynx. So this is the vestibule, which has skin, sebaceous glands and hairs, which are also known as vibrissae. Then whole we have the respiratory area, which is highly vascular and covered by the respiratory epithelium, that is also known as pseudostratified columnar ciliated epithelium with goblet cells. So in vibrissae we have hairs, skins and sebaceous glands. Then in respiratory area the lining is pseudostratified columnar ciliated epithelium with goblet cells, and then we have the olfactory mucosa, which is covered by olfactory epithelium to conduct the sense of olfaction or smell. Nasal sinuses. In body we have two frontal sinuses, ethmoidal sinuses, and then we have the maxillary sinuses. So these are the nasal sinuses which are covered by the respiratory epithelium. And then its function is to lighten the skull, to lighten the weight of the skull, and helps in the phonation of voice. So these are the sinuses which are used for the phonation of voice and to lighten the skull, and it is covered by the respiratory epithelium. Then the nasopharynx, also covered by respiratory epithelium, contains lymphocytes, mucus glands and the adenoids. So in this lecture you will have to basically learn the epithelium, like in nose which kind of epithelium is present, in nasopharynx what type of epithelium is present, what is respiratory epithelium. These are the major objectives of this lecture. Individually you should learn about the types of epithelium. The olfactory mucosa, which was present above the nose, the mucous membrane covering the superior concha at the roof of the nose. So it is covered with the pseudo-stratified columnar epithelium and has 3 cells: olfactory neurons, supporting cells or also called sustentacular cells, and the basal cells. These are the olfactory neurons. Olfactory neurons. These are the bipolar neurons, and they pass between the sustentacular cells. This is the sustentacular cell, this is the sustentacular cell. They pass between the sustentacular cells to the mucosal surface, and where they terminate into a small bulbous enlargement called the olfactory vesicles. It is a bipolar neuron passing in between the sustentacular cells and ending up in a olfactory vesicle. Then we have the sustentacular cells, which are tall columnar cells, relatively broad at their apices and tapering basally. They have numerous microvilli on their free surface with oval nuclei, and their most important function is that they provide nourishment, physical support, and electrical insulation to the olfactory cells. Then third type of cells, the basal cells, which are small conical or spherical cells that form a single layer in the basal part of epithelium. They act as a reserve cell that exhibit mitotic activity. So in the olfactory mucosa you have 3 types of cells. One is the olfactory neurons, which are bipolar, terminating into the olfactory vesicle. Then the sustentacular cells, which are the supporting cells, have microvilli, broad apex and tapering below, as nuclei is oval, and their function is to provide nourishment, physical support and electrical insulation. Then the basal cells, which are providing as the stem cells or reserve cells for further mitotic activity. Then this is the respiratory epithelium. As I've mentioned before, that the conducting part is covered with the respiratory epithelium. So what actually is respiratory epithelium? It is made up of pseudo-stratified columnar ciliated epithelium, also known as respiratory epithelium. The epithelium is pseudo-stratified columnar ciliated epithelium, known as respiratory epithelium, containing 5 types of cells: ciliated columnar, almost 30%, goblet cells, 30%, brush cells, basal short cells, the small granule cells. This is the cilia. This is the ciliated columnar cells, which constitute about 30% of the total cell population, and their function is to sweep the mucus here and there. The particulate matter is entrapped, and through their sweeping action it is passed away. So the ciliated columnar cells, the cilia, the goblet cells, which are also around 30%, they secrete mucin and give rise to mucus, which is a flexible gel-like material. The goblet cell, goblet cell, goblet cell. Here also we have a goblet cells. Then we have brush cells, which are almost 5%. These are the brush cells, which are cylinder columnar cells with numerous microvilli. You can see the microvilli, and their function is they act as a sensory receptor. Small granule cells, which are also almost 5%, and they resemble almost like basal cells. However, their cytoplasm consists of multiple granules. For example, this is the cell. It consists of multiple granules which are called as Kolchitsky cells. This is the Kolchitsky cells. So this is the small granule cell which resembles the basal cells, and finally we have the basal cell which act as a reserve cells or stem cells. So the respiratory epithelium is made up of pseudo-stratified ciliated columnar epithelium. It consists of 5 types of cells: the ciliated columnar cells, the brush cells, the basal short cells, the small granules Kolchitsky cells, and the goblet cells. This constitutes the respiratory epithelium, which will line the conducting part of the respiratory system. Then we come to the larynx. As soon as we come to the larynx, the laryngeal inlet comprises of a major structure known as epiglottis. So the epiglottis has a lingual surface and a laryngeal surface. Lingual surface is anterior and laryngeal surface is posterior. The lingual surface is covered by stratified squamous epithelium. Toward the base of the epiglottis, the epithelium gradually changes into pseudo-stratified columnar ciliated on the laryngeal surface. So the lingual surface is covered with stratified squamous non-keratinized epithelium, and as we move towards the posterior surface or the base, it is changed into pseudo-stratified columnar ciliated epithelium. The mucosa below the epiglottis form 2 pairs of mucosal folds, 1 pair and 2 pair, which are known as the vocal cords. The upper ones are the false vocal cords, which are covered with the typical respiratory epithelium. And what is that? Pseudo-stratified ciliated columnar epithelium. The upper ones are the false vocal cords. Then the lower part is covered with the stratified squamous epithelium. These are the lower folds which are covered with the stratified squamous epithelium, also known as true vocal cords. And here we have the vocalis muscle to regulate the tensions of the fold. Like when we speak, when we shout, when we cry, there is tension in the muscle which is created by this vocalis muscle. So here what is important for you is the epiglottis under the microscope. This is the laryngeal surface. And this is the pharyngeal part. So this is the posterior surface and this is the anterior surface. The epiglottis consists of a plate of cartilage covered on both sides by mucosa. This is a plate of cartilage covered by both sides of mucosa. The anterior lingual surface and the upper part of the posterior laryngeal surface are covered by stratified squamous non-keratinized epithelium. Till here it will be stratified squamous non-keratinized epithelium, and then the lower posterior half will be pseudostratified ciliated columnar epithelium. The glands are mainly found in the posterior surface, where they have irregular depressions, also called the pock marks. So these are some glands, maybe tubulo-acellular glands, also known as pock marks. Here it will be a bit cleared. This is the lingual surface and the laryngeal surface. The lingual surface is covered by stratified squamous non-keratinized epithelium. Then in between we have the elastic cartilage, its perichondrium. Then here is the transitional zone, where you can see the stratified squamous non-keratinized epithelium is now converting into the pseudostratified ciliated columnar cells with goblet. And this is the lamina propria, which consists of the blood vessels and few serous and mucous glands. So in this whole lecture, the most important thing for you is to learn about the epiglottis, and you will have to make a diagram of epiglottis in your practical journals. Other than that, you need to learn about the epithelium, like in nose, what kind of epithelium is present, in nasal, in nasopharynx, in nasal cavities, which type of epithelium is present and where. That will be more than enough. And then a little bit the histology of epiglottis only. So what we did is we talked about the respiratory epithelium. First of all, the respiratory epithelium, which is made up of pseudostratified ciliated columnar cells with goblet cell, which has 5 types of cells: the cilia, the goblet cells, the brush cells, the small granule cells, Kolchitsky cells, and then the basal cells. And then in the starting we talked about that respiratory system consists of the conducting part and the respiratory part. So this is the conducting part, which whole of it is covered almost by respiratory epithelium, which is pseudostratified ciliated columnar epithelium with goblet cells. Then the function of the conducting part was conduction of the air and to maintain the air conditioning through nasal hairs, capillaries, and respiratory mucosa. Then nasal cavity, we have a vestibule, the respiratory area, which was covered with the pseudostratified ciliated columnar epithelium with goblet cells, and then the olfactory mucosa, which was covered by the olfactory epithelium. So the olfactory epithelium consists of 3 types of cells. These were the olfactory neurons, then the supporting or sustentacular cells, and then the basal cells. Then the upper respiratory system consists of sinuses, frontal, ethmoidal, maxillary sinuses, which are covered by the respiratory epithelium, and their function is in the phonation of voice and lighten the skull. Plus the nasopharynx, which was covered with respiratory epithelium and contains lymphocytes, mucous glands, and the adenoids. The larynx, as soon as we reach the larynx, the first thing we come to interact is with the epiglottis. Below the epiglottis, there are 2 kinds of folds. The first one are the false vocal cords, and the second one are the true vocal cords. The false vocal cords are covered with the respiratory epithelium, and the true ones are covered by stratified squamous non-keratinized epithelium. And this is the picture of the epiglottis. You can make it. The lingual surface, which is continuous with the stratified squamous non-keratinized epithelium, and as soon as reaches the laryngeal surface, it transforms into the pseudostratified ciliated columnar epithelium with goblet cells. And here we have the lamina propria, which consists of blood vessels and glands. This is the cartilage plate, and this is what you have to draw. So this is the assignment. You will have to attempt these 3 MCQs, and you have to draw and label histological section of the epiglottis. So thank you. For any queries, you can always ask.